Hello, everyone. Today we are going to be going through our tuber lab, and you're going to need a few things to do this. So in Schoology, you will find that there is a link to a Google slideshow. It looks like this, and you are going to have a slide for you. And for each person in this class, you're going to pick a different slide. So if somebody has already started writing on the name, choose a different one. You're also going to need the amino acid reading that you can find. We're mostly going to be looking at this page here, but you can find this also in our lesson 9 folder on Schoology. So let's look at the materials that we have and let's walk through this. So the first thing that you're going to do is you're going to be using your green tuber, and it's this thing here, and we're going to be putting twist ties around it. And so you're going to fill out your slide with what we do, but I'm going to walk you through this. So here is my tuber. So I've got my green tuber. I have 6 of these gold twist ties, 3 silver, 2 green, 2 red, and 2 blue. So the first thing is I need to stretch this out, and you're not going to be able to see all of it, but I'm just going to start at one end. So this is going to be my amino acid sequence, and I am going to start at one end with a gold or a yellow twist tie. And I'm going to wrap this around a few times so that I can see it here. And then once you've got it wrapped a few times, you're going to twist it so that it locks it in place. And then you're just going to fold those ends out of the way so that they don't stab you. We don't want anybody getting stabbed in this class. All right, for the rest of them, you get to pick whichever order you want. You want them to be evenly spaced. So you're gonna stick the next twist tie about 2 fingers away from the last one. So let's say that I'm gonna choose blue. You choose whatever color you want. And again, we're gonna wrap it around. And twist. And then wrap that around as well. So I've done 2. You're going to put the rest of yours on. Again, any order that you want. I actually want your order to be different than mine. So I am going to do this, and then you are going to do it at the same time, and I'll speed this up on the video. Now that you have your whole tuber complete and you can see the order of the amino acids that you have put on there, you are going to change the color of the boxes on this slide in order to match the sequence that you have on your tuber. So mine goes gold and then blue. So I'm going to change this box That little thing indicates what it is. I'm going to change that to blue. And then my next one is gold. So I'm going to make this one gold. You see how this is done. You should be able to go through the entire thing because that's how many are on there. So I'll give you a chance to do that while I fill out mine. So now that you have your tuber made here with the correct number and whatever order you did, we're now going to use your codon chart in order to figure out which amino acids these represent. And the way that you're going to do that is you're going to look at your amino acid reading. So there are 20 different amino acids, and these amino acids are broken into groups based on their characteristics. So hydrophobic means that they do not like water, and those are going to be the yellow twist tie or the gold twist ties. And so if you have a gold twist tie, you can name it any of these that you want. Now, I'm going to suggest that your first one be MET because that is what the start codon codes for. But when you get to the next one here, you can pick any of these that are yellow. The clear or the silver twist ties, those represent these hydrophilic amino acids. And hydrophilic means that they love water. And so if you have a clear or a silver twist tie, you're going to pick one of these, except this one. This one is representing the types of amino acids that can form something called a disulfide bond. So that is represented by our green twist ties. So anytime you have a green twist tie, it has to be cytosine or CYS. Last but not least, we have our negatively charged and positively charged amino acids represented by red and blue. So as I go through here, my second one is blue. So I'm going to pick one of these that is blue. I'm going to pick LYS, and I'm going to type that in. And then I'm going to go to the next one, and I'm going to pick a yellow one. So one of these here, I'm going to pick PRO. And on and on. So give you a chance right now to fill in the rest of these based on your sequence. Okay, so now you should have an amino acid sequence that matches your tuber. We are now at the point where we can fold our protein into its specific shape. So your tuber here represents the amino acid sequence. And it's right now, it's a polypeptide. So it's made up of many amino acids and the peptide bonds that hold those together. But we need to fold this so that it can be in a functional protein shape, because the shape of the protein is what allows it to do its job. So if it doesn't have the right shape, if it's not folded correctly, then it won't be able to do its job. So taking a look at my sequence, I'm going to show you how to fold this, and you're going to fold yours according to your sequence. I'm going to fold mine according to my sequence. So to start off with, I'm going to focus on these gold or yellow amino acids. And we said that these ones are hydrophobic. Hydrophobic meaning they hate water. So what's going to happen is that all the hydrophobic amino acids are going to be folded inside of the protein so that they are protected from the water that is in the cytoplasm. So what I'm doing is I'm just bending my tuber, which has a wire in it, and I'm just trying to lock these in place in the center as much as possible, in the center of my protein. So those gold ones are now all locked in place. The next ones that we're going to look at are my silver or clear ones. These ones are hydrophilic, meaning they love water. So these ones are going to be in the outside of my protein because they are attracted to the cytoplasm of the cell. So I just want to make sure that those are sort of like pointing out as much as possible. Maybe they already are. So like mine was already pretty good. But maybe yours are a little bit more complicated and maybe they need more adjusting, and that's totally fine. Okay, the next thing that we're going to do is we're going to look at the reds and the blues because these are positive and negatively charged amino acids. And we know from experience with things like magnets that positives and negatives attract. So my red ones are going to be attracted to my blue ones. And this is where it starts to get a little bit tricky because you might have to do quite a lot of bends in order to make them touch. So you want those to touch and to kind of hold in place while trying to keep the rest of your protein following the rules. So we're still trying to keep our yellows in the middle, and as much as possible, we want those protected by other amino acids. So I'm actually going to fold this red one all the way around because then it's going to shield some of the yellow ones in the middle. So now that my reds and blues are about as close to each other as I can get, we now have our greens. And the green ones, these ones are really cool. You see, they have sulfur in them, and sulfur can form a really strong bond between sulfur and sulfur. And we call this a disulfide bond. So what's going to happen is our green ones are going to form that disulfide bond together. So green has to touch green. Now, in the end, I need to check to make sure that as much as possible, my clears are on the outside, my red and blue are touching, my green are touching, and my gold ones are in the center. Now, this is the shape of my protein, and my protein has a very specific shape based on the order of my amino acids. I have here another one, and you can see the shape of this protein is very different. It has the same amino acids, it has the same number of everything, but now it's folded differently because the sequence of the amino acids is different, and as a result, it's going to fold differently. So now that we have that, we're going to go back to our slides. And what I want you to do is I want you to compare your amino acid sequence to that of someone else's. So you're going to go into breakout rooms and do that. Write your answer here on how your amino acid sequences compare. And then I want you to upload a picture of your folded protein to this slide so that I can see what our folded proteins look like and so that we can see everyone's folded protein on this slide. Thanks, everyone. That's all for now.